All right, what we are going to do here is look at the structures, some of the structures within prokaryotic here and eukaryotic cells. We're going to look at some of their characteristics. We're going to look at similarities between them and differences between them. So first off is the name. All right, so prokaryotic, eukaryotic. So karyo. All right, so karyo in both of these cases refers to there being a nucleus. EU here means true nucleus. Prokaryote means without or before. So we're going to put without. That's one of the things that people will often refer to. They'll say prokaryotes don't have a nucleus, eukaryotes do have a nucleus. One of the things that then gets misunderstood is about what is a nucleus, what's the point of the nucleus, and then that. So the nucleus itself is a double membrane organelle. We'll just put membranous organelle containing DNA, which sometimes leads people to wonder about DNA. Like, do the prokaryotes have DNA? And yes, of course they do. It's just not inside a membrane-bound structure. Okay? So all these squiggles here, what they're representing are two things. The nucleus is actually a double membrane structure. Double membrane. There is, here, this is representing the inner nuclear membrane, this solid circle. And then you see the outer nuclear membrane is kind of like these pieces here. There's a little gap there, right? A little bit here, a little gap, a little bit here, there's a gap. What are those gaps? Well, those are actually spaces that then connect directly into this compartment. This compartment here is often referred to as just the ER, but it's endoplasmic reticulum. So the endoplasmic reticulum is the outer membrane of the two membranes of the nucleus in a way, but yet it forms a whole additional organelle that is even broken into two parts, a smooth ER and rough ER. We're not going to get into tons of detail on that. Essentially, there's a lot of things that happen in the ER. It's the place where membranes are manufactured, right, or made. It has enzymes that can detoxify chemicals. It can process proteins. So when new proteins are made in a cell, a lot of times where they are processed, where the folding takes place and chemical modifications take place, that happens inside the ER, or endoplasmic reticulum. And that would make sense when we think about the purpose of the DNA. DNA codes information for RNA that's used then to make polypeptides or proteins, and to get in their final form, it's the ER that kind of processes them to get them into that final form. All right, so all these big squigglies all around the ER, most of them, the ones that are connected are ER. There's also some here squigglies that I'm talking about that are disconnected, right? So there's this sac, 2, 3. Maybe I'll highlight them here a little bit extra so you can see the structure here, the structure here. You know, I'm doing, there's not just 3 of these. I only did 3 here. There could be more, could be fewer, but this is now a related structure called the Golgi apparatus. I may have run out of space, but it's called the Golgi apparatus there. It has some similar jobs as the endoplasmic reticulum, where it's also involved in processing proteins and the formation of these little vesicles. So you can kind of see here, I'm drawing these little circles, and they're actually been formed from some of these compartments of the Golgi apparatus, and then they might eventually, say, fuse to and become part of the cell membrane. That might be how proteins get embedded in the membrane in the first place, right? So the proteins are manufactured in this area with the endoplasmic reticulum. They're processed, they're embedded in membranes, and then the membrane piece actually is processed more and then eventually fuses with the outer membrane. So these are all, and there's other jobs as well, but these are all things that process proteins, package them up, transport them to the location where they're going to work, sometimes within the cell, sometimes in the cell membrane, sometimes outside the cell. So that's what all these little squiggle things here. And the point that I'm going to make beyond everything else is that they're made up of membranes. Okay, they're inner cell membranes, and they're providing surface area, not just on the outside surface of the cell, but now within the cell there are all these membranes. And that's going to be the key that we're really focusing on here. Now, coming back to the beginning where we were, we said the difference between pro and eukaryotic were nucleus, no nucleus. Okay. What is a nucleus? Membranes. Okay, there's membrane structure here, and it's associated even with other organelles, other membrane organelles. What's inside the nucleus is DNA. Both prokaryotes and eukaryotes have DNA. Okay, DNA in the form of chromosomes. Differences between them would be the eukaryotic, so I'll do little abbreviations here, chromosomes are linear, and then the prokaryotic chromosomes circular. So earlier lectures in the course, we talked a little bit about DNA structure. Okay, so remember we said that DNA has ends to it, referring to it at 3' and 5' ends. The 5' is a phosphate, the 3' is an OH group at 5'. And on the other end would be OH. Yeah, that one. So this 3' coming down here, then that's phosphate goes right. Double check it. That's linear DNA. That's what we find in eukaryotes. What happens in prokaryotes are that these ends join. So the DNA folds onto itself, and the ends are joined together, so it's circular DNA. So that's what we see here. Why is that? Well, why does a nucleus exist? Maybe we'll talk about that in the first place. Because the linear DNA, having these ends open, if they were open in free in the cytoplasm, there are enzymes who would come in and grab hold of and chew up the DNA and destroy it. So the DNA really wouldn't survive out in the cytoplasm very long before it was destroyed by enzymes. So it's protected inside the nucleus by these membranes, and these enzymes are kept separate. They're out in the cytoplasm. If those kind of enzymes are in the cytoplasm of a bacterial cell, that means they would destroy the bacterial cell DNA. So how do you protect these guys from being able to chew up the DNA? Well, if you join the ends together, then there's nothing for them to attach to. So part of the purpose of the circular DNA is that it's protective, and in this case, this came first. The circular DNA came before the linear DNA. Once the DNA could be protected, it could be opened up, and then there's other reasons why that might be an advantage, for those organisms. But they still have DNA. The DNA structure is the same. There are some eukaryotes with very few chromosomes. Most prokaryotes only have a single chromosome. Now you'll see what I drew here, though. I drew this extra little thing that looks kind of like that one, just smaller and a different color. It looks like maybe it's a second chromosome, but it's not. In prokaryotes, like this could be the cell chromosome. This structure is extra chromosomal DNA called a plasmid. That's the differences. Prokaryotes can have plasmids, which are extra chromosomal DNA. So it's their DNA separate from the chromosome, that they can be picked up from other cells. That's how they can acquire certain degrees of genetic diversity. That's how bacteria, say, for example, become antibiotic resistant. They could pick up genes from other bacteria who are antibiotic resistant. They can kind of transfer it to them. So almost by touch. Well, actually by touch, I'll talk about that in a second with some of the other structures called a pilus. That one cell can actually connect to another cell, make a copy of this information, pass it on, and say, hey, would you like some of this information? And here you go, and it'll give the genetic information, and the other cell will take it up and then become altered and changed because of that. And that kind of information's inside plasmid. So that's the just nucleus DNA stuff. Okay, so that's just the first part of this, really. And there's tons more with the molecular biology of them. Again, both similarities and differences, but that's not this lecture. It's more about the structure of it. Coming back to membranes, which is really the key thing that I want to like follow up on. Having the nuclear membrane and then these other membranous structures starts to create a lot of internal membranes. So that's one of the big differences, is that eukaryotes don't just have their cell membrane, they have many internal membranes. One thing that you'll usually hear or learn about bacteria is that they have no internal membranes. So, and I guess I'll talk about the size in a little bit. We're getting there. We're not quite there yet. Prokaryotic. Eukaryotic. So with the prokaryotic one, generally people will say no internal membranes. But I'm going to put a couple stars around that and even a little question mark. That's what you'll read most of the time, most textbooks and every place else. The reality is there are some prokaryotic cells who are very large, who are as large as, even larger than, many eukaryotic cells. And they have internal membranes inside them. And they are still prokaryotic because they technically don't have nuclear envelope around their DNA, and they have the circular DNA. And they have a whole bunch of other characteristics like a cell wall and things, which I haven't talked about yet, that make them prokaryotic cells. But they do have internal membranes. Most prokaryotic cells do not have internal membranes. Some of them will have, though, internal structure. So what people think of them as being super simplistic, but there's nothing much going on there. Like it's a little like membrane barrier, and then there's just a sac with just liquid inside it, and the DNA is floating around, and that's it. There's nothing else going on. There's a lot of very complex things going on inside prokaryotic cells, and there are many structures, even compartments, for prokaryotic cells. And so certain things have to happen, like the electron transport chain and pumping of ions and concentration gradients and diffusion, all within the cell. So how do you do that without compartments and chambers and membranes? And so they do have these things. It's just something that's often overlooked or oversimplified. So generally they don't. What the core characteristic they don't have is a nuclear membrane. And they typically don't have any other membranes. They can have structures that are kind of folds and things within the membrane here, things called inclusions and mesosomes, which not all of them have. Many of them do, and things called granules, which are not membranous, but they're where things can be stored, like concentrated packages of one kind of material, in a granule like that. And what I'm kind of getting at with this is the point of all these other membranes inside the eukaryotic cells. Cells need to store a lot of material. Right? And as they start to store a lot of material, what starts to happen? This is a cell, and you start to put stuff, let's just say anything inside the cell, maybe different sorts of things, different kinds of molecules. We're going to have water inside the cell and water outside the cell. And if the environment outside the cell is mostly water and very few of these other things, then water is going to want to move into the cell. And that's going to cause the cell to swell and become larger because more water is coming into it. And that could burst the cell, disrupt it. So that's osmosis. So that's a stress for cells. So cells are constantly under the stress of having to hold on to sugars and amino acids and all sorts of other molecules and ions, and all these things inside the cell. And that then makes this concentration difference greater and greater and greater. So one of the ways of combating this problem is, what if we packaged away a whole bunch of these things into little compartments and kind of move them out of the cytoplasm of the cell, so the cytoplasm doesn't have these things, they're all stored away in packages. Then technically the concentration in the cytoplasm of those things would be very low, right? And we wouldn't have as much stress from osmosis. So eukaryotic cells have many structures in them made up of membranes, all sorts of different structures made up of membranes. And that's what allows them not only to be, at the size now, bigger, but much more diverse in terms of size. So what you're going to read mostly in a book is the size of a eukaryotic cell, saying between 10 and 100 micrometers. So this little U, M, a little fancy U, so it stands for micro. So this is micrometer, or micrometer, but it's micrometer, so it's usually pronounced. So 10 to 100 micrometers, that's an average. We can have super tiny little eukaryotic cells that are just 1 or even less than 1 micrometer in size. That gets us to even the low end range of a prokaryotic cell, so very, very tiny. And then there can be monstrously huge eukaryotic cells. That's just an M for meter. A meter for a single cell. Nerve axon cell, and a giant squid, really large, large cell. So eukaryotic cells are generally classified as being bigger than prokaryotic cells. Prokaryotic cells being smaller. That's part of it. So size, so they can be incredibly large. But what's more dramatic here is the range in size. Okay, from a meter to a micrometer, incredibly diverse in terms of size. Why? Not only can they be bigger, but why can they be more diverse in terms of size? It's because of the internal membranes. All these internal membranes give them more surface. So not just the outer surface, but all the inner membranes count as surface. So more surface, more surface, more surface means you can create unique compartments. So instead of one big open room, you have walls separating bathroom from a kitchen, from other things, as opposed to everything in one space, which could be a problem in under certain circumstances, right? So here, the cells can compartmentalize jobs, specialized tasks, and store things away out of the cytoplasm where they're not worrying about osmosis because there's not as much osmotic stress because of it. So you have storage compartments like these. So some kind of vesicles, which would just be membranous structure for storage. Unique ones, like the structure here, doesn't matter. I'm just drawing little circles, right? But I'm going to call them by some of their different names. That would be a lysosome in my particular little model. That's a specific type of cellular organelle made up of membrane that has a really low pH, so it's really acidic inside here. Has lots of hydrogen ions, a bunch of them, and they're like little stomachs for the cell. They have also lots of digestive enzymes. So when cells are going to digest material that's either waste that they've created or something new that they brought in from the outside, that can happen in the lysosome, or associated by fusing with a lysosome called phagosome structure. They eat other things. So you can have that. We then have mitochondria. People call it a powerhouse of the cell, right? But it's a structure. It's a place where it's a double membrane structure, and it's where ATP is mostly made. So most of the ATP of the cell is made in the mitochondria for eukaryotic cells. Cells don't have a mitochondria. They have many mitochondria, right? And it varies by the type of cell. So muscle cells would have a lot more mitochondria than a fat cell because a fat cell isn't metabolizing as much, right? It's not as active as the muscle cell. So the muscle cell needs more ATP. It also has then more mitochondria. Cells have a lot of these all through it. So they have all these membranous organelles, all different kinds, right? The nuclear envelope, ER, Golgi apparatus, mitochondria, lysosomes, vesicles, all these sorts of things made up of membranes, all with different jobs storing different things. Inside the prokaryotic cell, very limited. Most everything is just within the cytoplasm. You can have very small structures that can store certain specific molecules to keep them together or to reduce their concentration in the cytoplasm. It's one mechanism that prokaryotes use. But that's pretty much it. They do have a space. So what I drew here is like two lines, all right? There's the inner line, it's this bluish purple line, right? And there's this outer line that's this more orange line. There's a space in between these two, the space here, called a periplasm. And my outer structure here is a cell wall. So almost all and this is kind of a big thing. Almost all prokaryotes have a cell wall. It is typically made up of the same material, called peptidoglycan. There's different ways it can be arranged, and there can be other molecules associated with them to be gram-positive or gram-negative, and there's a lot more complexity to it. But prokaryotic cells are going to have some kind of cell wall. This is for bacteria and archaea, generally made of peptidoglycan. Archaea can have different structures like a protein shell and other sorts of things, but that's typically going to be the case. The reason is they're going to be taking on a lot more osmotic pressure. The pressure in a prokaryotic cell of water pushing outward on the cell is equivalent to the amount of air pressure in a car tire. So that's the extent of the pressure pushing outward. A membrane made of phospholipids would burst and rip apart, right, with that much pressure. Just a standard membrane like this over here. So the only way that the cells stay intact is by having other mechanisms to mitigate osmosis, right, with active transport pumps and storage, and then also some structural support to hold back that pressure, which is the cell wall. So they need to have a cell wall, and that's the way you kill a bacteria, in a sense. Damage their cell wall, and the cells will pop often and burst. So certain antibiotics affect enzymes and proteins involved in the structuring of the cell wall. So by damaging the cell wall specifically, it will cause lysis of the bacterial cells and kill them. So because that's something that's important for them. Prokaryotic cells tend to be small. So in their size range, we have 1 to 10 micrometers. Now, there are exceptions. Okay? There is a bacterial cell that is about a millimeter, right? So that's 1,000 micrometers for a bacterial cell. You can see that's well outside the range of the average eukaryotic cell. That's actually 10 times bigger than the average eukaryotic cell. And that it's still a prokaryotic or bacterial cell. It does then have some membranes inside it that can then carry out these jobs, because that's going to be why it can't get big, because of the osmotic stress, really. So membranes help with that. So I have a separate lecture on kind of size and surface to volume ratio and all that stuff, which I go through in a little more detail. But that's kind of the concept of why can eukaryotes be bigger and have a lot of range in size, and also why are prokaryotes smaller, and also not just smaller, but have very small range. Generally, there isn't much difference in size between most of these cells. Like I said, there can be exceptions. The exceptions are very, very rare. The overwhelming majority of them all fall within this sort of range, and that's because they don't have a lot of internal membranes, so they can't carry out a lot of functions that mitigate the osmotic stress. Another similarity, which is not drawn too well on here yet, are ribosomes in the cytoplasm. So a ribosome is another type of cellular organelle, but it's a unique organelle because it's not made of membranes. So prokaryotes actually have organelles in their cytoplasm, which are ribosomes. And eukaryotic cells have these ribosomes. Many of them get attached to the endoplasmic reticulum. This is what we call the rough ER. It's called rough because if you look at it, surface looks like it has all these little studded structures attached to it, and those are all ribosomes. So there can be ribosomes free floating in the cytoplasm in eukaryotes, but a lot of them get attached to this endoplasmic reticulum because they're making the proteins and they're pushing the proteins back into the ER lumen where they get processed and go through all the rest of this. Other similarities, and this kind of goes in the similarity and difference kind of category. So it's a little bit of both. So having a flagella, so some, not all bacteria have flagella. Some eukaryotic cells have flagella. Not all eukaryotic cells have flagella. Flagella allows the cells to be modal, so they can move around. They can swim in water or move through tissues, all these sort of things. Or if the cell's anchored to a place, the flagella can be moving to create flow or water current across the surface. Right? Some choanoflagellate cells in animals, like in sponges, that's how they feed. They create water current with these flagella from these collar cells, and then they capture the food and phagocytize it, so they actually use the flagella to create water current to catch food, essentially. So there's all different reasons why they might have them. The main thing is, though, is that the difference is flagella structure. Very different, right, between them. So the flagella structure in a prokaryotic cell, if they have it, like so they do not all have these structures. First off, it's solid protein called flagellin, and it moves in a spinning fashion. So there's a motor protein embedded in the cell membrane and cell wall that actually causes this protein whole series of very complicated little motor structure, actually, and it like spins, and then this structure sticks off it and it starts to whip around and it moves like a corkscrew. And so the flagella of a prokaryote is spinning and spinning and spinning around like this. A eukaryote, very different structure. Flagella are kind of important among eukaryotes in that they really do unite all the eukaryotes, from the single-celled eukaryotes to multicellular organisms. The type of flagella that we have on our cells is the same as the type of flagella a euglena would have, which is a protist, a different kingdom. Plants cells can have flagella. Some plant cells have sperm that actually swim around and fertilize eggs, right? And they have a flagella that's just like this. Some fungi have chytrid, these are the type of fungi, have flagella, and they're single-celled fungi that swim around with a flagella. Some animals that are have cells lying within their bodies, or some part of them have flagella. So, and they're always the same structure. So there's 9 of these doublet tubules with 2 at the center, kind of like a wheel. And the way in which it works is that there's a sliding mechanism, right, that causes this flagella structure to just kind of bend. And it just kind of like it shortens on one side and it bends, and then it gets pulled back and it gets shortened on the other side, and the side extends and then it gets pulled back. So it really is just a windshield wiper like movement for a flagella. The longer it gets, then the more wavy it gets in its motion or movement. So technically, cilia and flagella are really exactly the same in terms of structure. It's just that the cilia are usually very short and they just move back and forth really fast, and cells can have many of them. They can be completely covered with the cilia. When they have flagella, the flagella are long, and then they tend to only have one or very few of the flagella. Bacteria don't have cilia, right? They can just have a flagella. And if they have a flagella, they could have one, they could have 2, so they could have one at each end. Interesting. They could have a tuft of them, so actually you could have multiple. This is actually more common. There's multiple flagella all coming out of the same area. That's a bit more common. And so, but they're all structured the same as each other. But not all of them will have flagella in this sense. Bacteria can produce some other structures as well. that are different than the flagella, but they can be used in some sort of similar ways. So they can produce something called a pilus that's unique. So prokaryotes can produce something called a pilus. And what this pilus allows them to do is, let's pretend this is another bacterial cell over here. They can extend part of their cell membrane and create a tube like this. And they could use that tube to actually get a copy of a piece of DNA. Usually it's from a plasmid like this. So a little piece of DNA is copied, or the whole, actually one strand of this plasmid is copied, and then it can get passed along. So it's like a bit of DNA. It can get passed from one cell to another cell through this pilus structure. That's unique for prokaryotes. Eukaryotes don't form pilus. The pilus can be used for just simply attachment, to allow one cell to attach to another cell, not pass genetic material. This is called a sex pilus, because it's passing genetic information. Another pilus is just for attachment. Structures called fimbriae and pili are pretty much the same. They're made up of the same protein called pilin, and they can have similar jobs, or they connect to different things. They can connect bacteria, or bacteria to your cells. Your body's covered in bacteria. The bacteria inside your body, most of them are attached by extensions that come off the cells called fimbriae. And essentially they're the same thing as a pilus. They're made up of the same material. They don't actually connect to the interior of the cell. They just stick themselves to the surface and have special proteins that kind of bind and hold on. I think all the main stuff I needed to cover in this, we have gone through. So we have some big differences between prokaryotic and eukaryotic cells, to summarize. First one is just nucleus. Eukaryotes, true nucleus. Prokaryotes, no nucleus. Second one is generally size, and that's related to the membrane. So prokaryotes usually smaller, eukaryotes typically bigger. Eukaryotes not only bigger, but more diverse in terms of size. Why are they bigger? Why are they more diverse? Lots of internal membranes, lots of compartments, lots of surface area. It allows them to be much more efficient and handle that osmotic stress. Whereas prokaryotes can only get up to a certain size, and they can't handle the osmotic stress. Their efficiency is tapped out, and then the cells could burst and die. They're limited in their size. They both have DNA, but their DNA is slightly different in the way it's organized into linear strands versus circular ones. Prokaryotes typically have just a single chromosome, whereas eukaryotes typically have many. I just drew one little DNA there, but they usually have many, many chromosomes, different for different species of organisms. Lots of internal structures I've talked about here with eukaryotes, different organelles like ER and Golgi. You should be familiar with just the basic term name definition of what each of those organelles are and what they can do. They can both be motile, have flagella, but they're made up of totally different materials, and they function in very different sorts of ways. Hopefully that gives you a good idea of what is a prokaryotic cell and how is it similar to, in some ways, but different from a eukaryotic.